TDP-43 does many things, as we know, and it is certainly well appreciated that TDP-43 regulates the like thousands of transcripts, both at the mRNA level, non-codings, microRNAs, etc. And TDP-43 can bind to these various pre-mRNAs, RNAs, both within the nucleus, regulating distinct steps of mRNA and microRNA processing. Also, TDP-43 shuttles into the cytosol, where in neurons it has been reported to regulate RNA transport processes under normal situations or stress granule formation under pathological situations. So since TDP-43 does so many things, it is obvious to assume that the key to the understanding of the molecular mode of action of TDP-43 must be within distinct complexes. In contrast to the enormous amount of data available on the regulated transcripts, the protein interactions are not so super well studied. So this is a relatively old study in which you can see on a gel filtration trace that TDP-43 actually forms two distinct peaks. Subsequently, Kubahara, this was the microRNA regulation paper, you remember, where they could identify TDP-43 being present in distinct complexes containing the microprocessor complexes. So based on this rationale, we figured that it is not enough to know all the transcripts that are regulated by TDP-43, but we must understand the protein embedding in which TDP-43 acts. And for this purpose, we went to perform a yeast-to-hybrid screen to gain insight into potential interactors of TDP-43 that might expand our knowledge about the mode of action of TDP-43. I will try to be self-critical and discuss also the downsides of this project while I'm going on. So when we first started out with wild-type full-length TDP-43 as a bait, we found nothing at all that would make any sense whatsoever. Only the usual, ribosomal proteins, which are known false negatives. And in parallel, we wondered if we could enrich for protein interactors for the CTF, like a construct which would resemble the pathognomonic TDP-43 fragments. This construct gave, under high stringency, about 10 potential interactors, of which we were able to clone 7 and validate the interaction in co-immunoprecipitation upon co-transfection in HEC293 cells. Curiously, or maybe not, since we used the CTF as a bait, the interactions were generally stronger with the CTF compared to the full-length interaction. In one case, it was pretty dramatic. Look, this is the ubiquitin-conjugating enzyme UBE2E, where we hardly saw any interaction with the full-length protein under these conditions, where we are IPing the target with the tag, and we're probing for a cherry tag TDP-43. If we do the converse assay and IP TDP-43 and probe then for the tagged interactor, we can see the interaction with full length. So some of these potential TDP-43 interactors are either indirect binding or they are engaged in a larger complex which requires optimal conditions for co-immunoprecipitation. This list of interactors contains interesting proteins. One here, the RBM45, has been published some time ago now to co-aggregate with TDP-43 in pathological inclusions. The mediator complex is interesting in itself, being a regulator, master regulator of transcription, particularly in the connection between distal enhancer elements and the core promoter. RAC1 may be also interesting in terms of ribosomal biogenesis and RNA transport. LSM6 is a component of an mRNA processing complex, both in the nucleus and in the cytoplasm. So these are basically providing hints for future analysis. Pragmatically, we focused on the ubiquitin relevant proteins. We found one D-ubiquitin, a dub, which is called USP8 or UBPY. And we found this E2 ubiquitin-conjugating enzyme UBE2E3. Basically our model hypothesis, initially our working hypothesis, was that TDP-43 is ubiquitinated. You see that in patient samples. And that the UBE2E3 is a major regulator of the ubiquitination of TDP-43, while the UBPY is dubbing it. So here's the data. So if we are overexpressing TDP-43 with UBE2E3 family members, mind you, UBE2E3 has 3 members of the class, 1, 2, 3. And all 3 members are, even in plain lysates, capable of causing TDP-43 band shifts. If we are co-overexpressing a his-tagged ubiquitin, which allows us the pull-down of ubiquitinated protein from highly insoluble fractions, which is an important step, then you can appreciate that UBE2E3, UBE2E2, and UBE2E1 all mediate ubiquitination of full-length TDP-43. In contrast, UBE2EN, this is the K63 linking ubiquitin-conjugating enzyme, hardly leads to the ubiquitination of TDP-43. UBE2EC is completely ineffective. Perhaps more importantly, if we are mutagenizing the ubiquitin activating site, the ubiquitin binding site on UBE2E3 by converting the thioester forming C135 to serine, then we completely abolish the ubiquitination mediated by UBE2E3, suggesting that we're not dealing with overexpression artifacts, but really catalytic activity is necessary to confer the UBE2E3 mediated ubiquitination of TDP-43. Now, here it gets a little fishy, and Tito was sort of already starting to allude towards it. So usually, when you are overexpressing a E2 enzyme, what should happen is that you have polyubiquitination, and if you have more than 4 ubiquitins attached, that is a targeting signal for proteasomal degradation. But as you could see here easily, is that the steady-state levels of TDP-43 are not reduced. So we're not talking about the classical ubiquitin targeting ubiquitination, but something else. And really remarkably, when you look at the ubiquitinated TDP-43 species, you will find them exclusively in the highly insoluble urea extracts, indicating that the type of UBE2E3 mediated ubiquitination is not a clearing one, but somehow it converts TDP-43 into a highly ubiquitinated, highly insoluble species. The converse experiment is important, of course, so still one can always argue that we're dealing with overexpression artifacts. So in this setup here, we did the same thing. We transfected with full-length human flag TDP-43 plus his-tagged ubiquitin, and now we knocked down the endogenous UBE2E3. So if this is the control situation here, you see that you can force TDP-43 to be ubiquitinated, and when you knock and the scrambled situation, and if you're knocking down the UBE2E3, you see a great reduction in ubiquitination, suggesting that the endogenous UBE2E3 does contribute to the ubiquitination of TDP-43. When you retransfect wild-type UBE2E3, you can restore the ubiquitination, but not so if you are trying to rescue with the catalytically inactive variant. So I guess it is safe to conclude that UBE2E3 or UBE2 family member activity contributes to the ubiquitination of TDP-43, which is correlated with a shift of TDP-43 into the pathological insoluble fraction. So the next obvious question is, how do TAR-DPP mutations influence this process? And we have been looking at 15 of the 40 published TAR-DPP mutations, and none of them is really conspicuous. Notably, all mutations in the glycine-rich domain or prion-like domain or low-complexity domain, as whatever you want to call it, are completely unconspicuous in all regards we have ever looked at. The only two TDP-43 mutants that are more fun to work with are the ones that happen to be outside of the GRD. D169G tends to be expressed a little less in all experiments we do. And the only, in this context, as far as ubiquitin is concerned, really absolutely unique mutant is the K263E mutant, which is a rare variant found in an Hungarian kindred, which interestingly does not correlate to motor neuron disease. This is one of the super rare FTD mutations of TDP-43, and in the CNS it has wider functions. As you can see, even upon expression alone, K263E, TDP-43 is a little bit more ubiquitinated, and when you highlight this with pull down of ubiquitin, you see that K263E is whoppingly ubiquitinated, completely distinct from all other mutants we have ever looked at. Why is that so? I don't know. This here is the NMR structure of the TDP-43, and this is a zoom in into the RRM2 where the K263E mutation is localized. And as you can see, the K263E residue is a very, well, almost the last one that forms hydrogen bonds with bound RNA. So K263E is clearly altering the RNA binding capacity. Why the absence of RNA binding is related to more nuclear ubiquitination, I don't know. But it is likely to assume that if you have excessive ubiquitination, you might disrupt the dimeric structure of TDP-43 within the nucleus at that site, and you alter basically the conformation such that it becomes ubiquitinated more by the nuclear ubiquitin-conjugating enzyme UBE2E. And this is the data. Can you, Mr. Chairman, Aaron, can you remind me of my time because I keep losing track? 5 more minutes, perfect. Okay, so here, this is the experimental results with the KE mutant. So as you can see, if you're overexpressing wild-type UBE2E3, the already conspicuous ubiquitination is dramatically enhanced, whereas if you overexpress the catalytically inactive UBE2E3 variant, you see no ubiquitination. Conversely, if you look at the normal ubiquitination of the K263E mutant and you're knocking down the endogenous UBE2E3, you can see that it is significantly reduced. Now if I move on to the DUB, to the de-ubiquitinating enzyme, it is, well, coincidentally quite convenient that the KE mutant is so excessively ubiquitinated, so it is a nice tool to study the ubiquitin removal by DUB enzymes. So basically, we have done the same thing here. So we start with the basal ubiquitination of K263E TDP-43, and now we co-overexpress wild-type catalytically active UBPY. And you can see in the presence of this de-ubiquitinating enzyme, the ubiquitin chains are removed. However, if we are co-overexpressing a catalytically inactive point mutant or a construct of UBPY in which we deleted the catalytic domain altogether, in both cases, the de-ubiquitination does no longer occur, suggesting that UBPY is a DUB for a TDP-43. Now with UBPY we have a little bit more of a conceptual problem because unlike UBE2E3, which is nuclear and everything is fine, that's where it's localizing where TDP-43 is localizing, UBPY in fact is not nuclear at all. So the job of UBPY usually is to regulate endosomal trafficking of receptors, and that's why you will find UBPY, both the transfected variant as well as the endogenous one, near the plasma membrane in endocytic vesicles and in these blobs here, which is Golgi. We confirmed this with GM130 staining. So nevertheless, I still believe that there is a chance for TDP-43 to interact with the cytosolic UBPY because, as we know, and here this is shown in our cells, that TDP-43 with long exposure does visualize the shuttled cytosolic material, and although there is clearly UBPY alone endosomal vesicles, there is some overlap with TDP-43. So we reasoned, according to the multiple hit hypothesis, if we were predisposing cytosolic relocalization of TDP-43, we would enhance the chance of its interaction with UBPY, and it would become a better substrate than the normal form. And this is indeed what we see. So in this case here, we used either a GFP-tagged CTF, which is localizing throughout the cell because it is so small. And we also took a flag-tagged NLS mutant, originally coming from Virginia's lab. Yes, indeed, I hate, or well, we can point it either way. If you really want to see an aggregate, then you fuse a cherry onto your target because cherry makes it aggregate so nicely. So it's like a really cheap trick to get aggregates. But of course, the chances are high that you're dealing with artifacts, which is why we why we also prefer small tags for our aggregation studies. So anyway, if you now take the NLS mutant, you will see that the co-immunoprecipitation with UBPY is much, much stronger, obviously due to the fact that we're relocalizing it to the proper compartment. To go through the data real quick, indeed, TDP-43 mutant, both the cherry CTF as well as the arguably more credible NLS mutant flag tag, you can see that if you force its ubiquitination, the expression of UBPY reduces mutant TDP-43 ubiquitination, while the overexpression of the catalytically inactive point or deletion mutant failed to deubiquitinate cytosolically mislocalized TDP-43 mutants. This is my last piece of data. So Tito already introduced the Drosophila model. Here, this is flies from Aaron Vogt in Aachen. Here, TDP-43 GFP is driven by the GMR driver in the Drosophila retina, and you can see the progressive phenotype, which is well known, and Tito explained it before. If we are expressing UBPY RNAi, but also by the GMR driver in the eye, you can see that there is a minimal or a very, very mild rough eye phenotype. And if we are combining both, you can see that at day 1, you have an enhancement of phenotype which which aggravates with time. Here, this is like one of the rare examples of flies that live to this time point to indicate the severe enhancer effect of UBPY knockdown. So if you are preventing in the fly the removal of pathologically associated insoluble TDP-43 ubiquitination, then you are obviously creating an enhancer effect. And to document this biochemically, again, conveniently, Tito explained the assay. So if you look at the fly heads, for us it's 1 to 5 heads are enough, then we can see that in an age-dependent manner, you have a reduction of the normal TDP-43 reflecting the degeneration. But if you're looking at the ubiquitinated species here, you will see that in an age-dependent manner there is an increase in polyubiquitinated insoluble TDP-43, and in the absence of UBPY there is a great enhancement in the amount of insoluble, evidently polyubiquitinated TDP-43, consistent with the idea that the shift of ubiquitin-mediated insoluble TDP-43 contributes to neurodegeneration in the overexpression model in Drosophila. So far, so good. There is a few remarks well, okay, no more concluding remarks, okay, so then only the good news. TDP-43 is ubiquitinated. It is positively regulated by UBE2E3, negatively regulated by UBPY, and it could be one of the hits in the multiple hit hypothesis of FTD pathogenesis. Okay, so thanks for your attention and your patience.